third lesson, evolution and diversity, by finishing the second lesson together with the final practical. Okay, so class updates wise, this Thursday we are beginning the class with our small paper, right? That is going to be the first paper that you are going to get. It is going to be from the first lesson as well as the entire second lesson. Very short paper, 15 MCQs, one structured essay, yeah, 1.5 structured essay, okay, and one essay question is going to be there. Small 1.5 hour paper. Okay, so it's Monday, you have Tuesday, Wednesday, and also Thursday morning to study for that. It's nothing serious, but I want you to have the exam exposure, start writing some papers from the beginning itself so that you can understand what you need to improve, how to write an essay, right? Then I'll be marking that and giving it back to you. Okay, so for Saturday people, it will be happening on Saturday. And for Thursday people, physically you can write the paper at Hedveni Gambo, and online people, the paper was delivered to people who were had enrolled at the website, and together with our new tutes, okay, for the evolution and diversity, the brand new tutes are on the way or already in your hands. Okay, this is the question tute. It is going to have only classified past paper questions and a few diagrams, Hari. And this is our main tute, that is the diagram tute, and we are going to go ahead by making our, let's say, short notes and our own notes as we go ahead. This will be I only got the unit 1, 2 and the paper. How is that possible? You didn't get the unit 3 tute? Because it was supposed to be in the same tute package that where we sent the paper. Okay, so the paper envelope you are not supposed to open until the exam day. If you are a Thursday student, you are not supposed to open it until the Thursday 3 p.m. And if you are a Saturday student, you are not supposed to open the envelope with the paper until Saturday, where you are supposed to join our Zoom live and then write the paper in the Zoom class. Okay, so don't open that with the confusion. Okay, then the other thing is the hotline numbers are available. Okay, the hotline numbers are available. There are 2 class hotlines to handle any issue related to tutes, website enrollment, coming to classes, if you missed any classes, if you want to get any recording there from a missed single day, if you want to get a recording. Online people, you don't have that issue. No, it's available on the website. Physical people, you can request recordings from the hotlines. And online people, any tute issue, any payment issue, there's a separate website person that is allocated to you. He's almost always there to solve all your issues. So those numbers are available to you all the time so that you can solve them. I'm again and again sending those numbers, so you better save those numbers so that you can use them at your convenience. The issue, okay, so the hotlines are there to solve all your problems as soon as possible. Okay, anyone so hands up if you got the third lesson tute. Hands up if you got the third lesson tute. Okay. Anyone who paid the fee and enrolled this month and didn't receive it yet, okay, before Thursday it will be there, because with the postal service there could be a slight delay, but it will be there before Thursday or before next Saturday definitely. Okay, so don't worry about it. If it's not there, you can contact the hotlines and also let them know as soon as possible if you didn't get the tute yet, okay, before the Thursday's class or before the Saturday's class if you are a Thursday or a Saturday student. Right? And so today is up to speed sessions final day. Up to speed sessions final day, and so right on time today I am finishing everything that we were doing in the theory, we have completed in the theory class before you got your O-level results. Okay, so the main goal of this program was to quickly help you complete the tutes before, so that you have a completed tute as you are joining our regular theory classes. Okay, so because otherwise you have to watch some at least 12 recordings. Okay, 12, no, more than 12, yeah, around 15 4-hour recordings you have to watch. So what I did here was within 6 hours, 6 recordings including today, you can complete the tutes, and you can have, so the second and first lessons, most of it that was done before you join the class will be covered. Okay, so today is the final day. We have a small bit of mitosis remaining to be done. Small bit of mitosis remaining to be done, and then we talk about some cancers and goals in plants. Then we complete meiosis, and if we have time, we are going to move on to a little bit of introduction to metabolism and some energy relationships. Okay, and okay, so enzymes day 1 recording will be sent to all the groups. Enzyme starting 1 will be sent to all the groups, so that you can watch it before the exam and start preparing. The main thing I want to emphasize is, this is, don't worry excessively about the paper, but there will be top scorers. I will be giving out top scorers, physical top scorers as well as online top scorers, top 10 probably. Okay, so keep that in mind. It's good to have, good to be on the top from the beginning. That doesn't mean, so everyone can be on the top. This is another, there's almost another 2 years remaining in this journey, Harry. Almost 2 years remaining. So there's plenty of time for you to come to the top. Okay, so now 26 people today themselves, my 26 batch, they went to the exam and the MCQ paper is over. So with that, there's only 27 batch in front of you. Okay, so then it's you guys. Okay, so keep that in mind. You have only exactly 2 more years. So this is 26 August, 7 August, 8 August, only 2 more years. We started this with 2.5 years almost, but now you have exactly only 2 years remaining, and in December and in January the 29 people are going to come. So you are going to become the second years. Okay, so that's how time goes. So slowly we have to get into the rhythm of A levels. I'm going to guide you throughout the process. When are we starting paper classes? Hurry, now listen. Paper classes will be started for you guys in 2 steps. Paper classes will be started for you guys in 2 steps. One, we will be starting an online only paper class where we are discussing 1 full paper per month, 5-hour full paper per month. That paper class will be started as soon as we finish the 3rd lesson. Okay, so that means in another 3-4 months time we'll be starting that paper class. Then it will continue until the 27 people are done with their, writing their exam. That means in 27 August, I'll be starting your proper paper class. Proper paper class means it's a physical as well as online hybrid. So as our normal theory class, there will be doing day papers where I'll be going from the beginning of the syllabus with day papers, small papers. Then also again, per month there will be 1 full paper in that paper class as well, so that it will be covering, that full paper will be covering everything that you have done up to that point, all the theory. So you can understand, no? So that paper class will be going from the beginning again through question papers, and at the same time we'll be doing full papers every month, so that you can see how your marks are improving every month until the exam. That's the paper class program. I just started that for 27 people. In another year's time, that proper one will started for you. But as soon as we are done with our 3rd lesson, we'll be starting our 1 full paper per month online only paper class. Hari? Okay, so I'll keep you all updated when we are starting everything. Right, anyway, right, shall we come back to today's session? We have to finish mitosis first. Okay, so we talked about our cell cycle. The cell cycle is simply the life cycle of a cell, no? How a cell lives its life. right? So from the day it divided, right? So you can see this is the point where it was born from its mother. So then onwards, it's going to just live a normal life until it itself divides. Then you were born from your parents and you live a normal life until you have your own children. No? So the cell cycle is simply that. So it has how many phases? You actually have the big one as interphase, as well as the phase where the cell division happens is called as the M phase. So the interphase has three sections as G1, S and G2. Okay, we talked about what's happening in the G1, S and G2, right? So after the cell has prepared with all these G1, S and G2 phases, it is going to enter into the finally the dividing phase, that is the mitosis, right? So this is the cell at the end of the G2, right? So you can see what are the things. I will quickly go through it's happening here. So in cell division, we are only going to focus on what is happening in the nucleus. Sorry, what is happening in the nucleus. That's all we are going to focus on, right? So there you can see we are only focusing on the nucleus, so all the other cellular organelles are ignored, right? And you can see you need an apparatus, and that means like to control all the chromosomes that will be produced, you need an apparatus. That apparatus is actually called as the spindle. That spindle is given here. I think this yellow color, all these structures is the spindle, right? So if I'll color it over with red, the spindle has two centrosomes. We are calling this the centrosomes. They are composed of centrioles, right, arranged at 90 degrees, and they have very long microtubules that are extending forward. There are two types of spindle microtubules. That means there's one type of microtubule that is connecting to this chromosomes. Can you see? They are the microtubules connecting to chromosomes. They are called as anyone? The microtubules that are connecting to chromosomes, they are called as what microtubules? They are known as what microtubules? The microtubules that are connecting to the chromosomes of the spindle, yes, they are called kinetochore microtubules, because we found out here also. You can see this part in the chromosomes, in the two chromosomes where the microtubule can hold on. It's a special protein, it's called the kinetochore. So if a protein is attaching to that kinetochore, if a microtubule is attaching to that kinetochore protein, then it's called as a kinetochore microtubule. Then there's another type of microtubule that are not connecting with the chromosomes at all. They are interacting with the same same microtubules from the opposite pole. So you have red and blue. That's what I use two colors. So the blue people are known as non-kinetochore microtubules. No? Very easy to remember. If you are connecting to a kinetochore, you are a kinetochore microtubule. If you are not connecting, you are a non-kinetochore. Okay, simple theory part, right? So where were we? We were just at the phase where the chromosomes are connecting, that is our pro-metaphase. They are being connected to the kinetochore microtubules and they are being moved to the sort of the center of the cell, so that the cell division can, cell division means the separation of the chromosomes can actually begin. Okay, so we'll continue from metaphase onwards. We'll continue from metaphase onwards. Okay, we'll continue from metaphase onwards. Let's come to the resource book, everyone. Let's come to the resource. Okay, all right. Where are we? Page number metaphase, page number 41. Okay, we'll quickly get this over with. Okay, right. Metaphase, what's going to happen? Centrosomes reach the opposite poles. Centrosomes have reached the opposite poles. So you can see the yellow color centrosomes are completely found in the opposite poles. Okay, because you need one at each pole so that they can pull the chromosomes apart. Right? If I can quickly remind you, what does the chromosome look like? Our chromosome actually has two exact copies. Our chromosome actually has two exact copies. What are these exact copies? So this is one copy. This is the other copy. They are connected at a part of their structure that is called as centromere. They are connected almost not exactly at the center, but they are connected at this centromere. So can anyone tell me? So these both of them form a chromosome. Correct. Can anyone tell me what do you call these individual copies? What do you call these individual copies of this chromosome? What do you call this? Individual copies? Okay, not halfway there. Halfway there. Halfway. What is the full term that you used to describe? Yeah, sister chromatids. They are chromatids, yes, but they are also sisters. More like twin sisters. Sorry, more like twin sisters. Because they are exactly identical, exact copies. Why are we making exact copies? Because at the end they will have to be separated so that they can be given to the two cells, right? Because when you, if you have, I mean, if your parents have two children, they might have to separate exactly your land in the middle and give them exact portions. Sometimes otherwise siblings atare there will be problems. So similar to that, the mother cell has to copy her DNA and then give them to the two daughter cells that will be produce exact copies. Exact copies. That is the main thing about mitosis, is that you give out exact copies. DNA doesn't change at all. It's just cells are like exactly like cells photocopying. Okay. So metaphase. What else? Chromosomes have arrived at a place. The chromosomes have arrived at a place. Chromosomes have arrived at a place called metaphase plate, which is located equal distance from each pole. Right. Can everyone please label this? There's a line. There's a line going across, dotted line. That is actually equidistant. If you can say this is the equidistance, these are same lengths. Okay. So what do you call this? What can you write in this box? Right here, this box label, it is the metaphase plate, which is an imaginary line. Then it's an imaginary line. So if you take a look at our Earth, there's nothing called equator. No, it's an imaginary line. Equator. You can't see a dotted line on the real Earth. No? So if you take a cell, there is nothing like a metaphase plate. It's an imaginary equidistant point. Okay. Keep reading. Resource book we are reading. Okay. Centromeres are located in the metaphase plate. At the end of this phase, each chromosome of the cell get attached to the microtubule at their centromere and aligned at the metaphase plate. So this metaphase, now listen, is like before a drama, before a stage drama, you have to set the stage. All the background should be there, all the people who are in this position should be in their correct positions, all the actors. Okay, so before you start a stage drama, you have to set the stage. So the metaphase is actually like before the drama begins, the stage is set. The drama is actually this: chromosomes, chromatids being separated. That is the drama. So the drama is about to start very soon. So metaphase is the stage is set. Okay. So we'll write down here in the tute. We'll write down what's going to happen in the metaphase. Hari. So you can write within underneath it exactly. Hari. So you can say first one, centrosomes reach, write with me, centrosomes reach opposite poles. Centrosomes reach opposite poles. Second, centromeres, write with me, okay, then say we can save time. Centromeres of all chromosomes, centromeres of all chromosomes are arranged at the metaphase plate. Centromeres of all chromosomes are arranged at the metaphase plate. Put a star mark and you can write down metaphase plate is located equal distance from each pole. Okay, metaphase, simple phase. Huh? Okay, quickly write this down. Okay, right, clear. I'll at the end of this I'll be again showing the software, I mean our sort of animation, so that you can have an idea about the overall final picture, what's going to happen of at the end of a mitotic division. Okay, done. If you wrote with me, you can save time, Harry. Right. Shall we move on to the drama? That means what's going to happen, so that we were preparing, our cell was preparing for this phase for a long time. That means the separation of our sisters. The sisters are about to be separated from their centromere, and they are going to go into individual, I mean go to the opposite poles first, go into opposite poles first. That is the sort of the main event in this drama. That means where the sister chromatids are separated. So let's read. So we are in anaphase. Okay, let's read. We are in anaphase. What's going to happen? Sister chromatids are separated at the centromere. Microtubules attached to kinetochore get shortened. Okay, sister chromatids are separated. That's very important. Can you highlight? Sister chromatids are separated at the centromere. So the centromere is like a bit of glue. That means they are connected by the centromere. Now the glue will be sort of dissolved and they are separated. Microtubules attached to the kinetochore shorten and pull the sister chromatids towards the opposite poles. Can you highlight? Microtubules attached to the chromatid. So I use the red color to indicate kinetochore microtubules. No? So you can see here, these are the kinetochore microtubules. These are the kinetochore microtubules. And they are going to shorten. They are going to shorten. So they are going to sort of pull the sisters on this direction. They are going to pull the sisters in this direction. On the other side, the same thing is going to happen. The other side, same thing is going to happen. So the sisters are now separated. It's like once the sisters got married, their husbands are pulling them to opposite poles, right? Separate houses. Okay, right? So now the sisters are leaving each other. So that's one thing. So the kinetochore microtubules are going to what? They are going to shorten. What's going to happen to non-kinetochore? Highlight. Cell elongates as the non-kinetochore microtubules lengthen. Cell elongates as non-kinetochore microtubules lengthen. Right? So the blue non-kinetochore microtubules, they are going to do something opposite. Then these are the non-kinetochore microtubules. So these guys are going to elongate. Okay, so they are going to sort of push each other like this. They are going to push each other like this, and that makes the cell get long. That makes the cell get long. Because why? At the end, you can see in the next step, the cell is going to be separated into two cells at separate into two. So in order to happen that to happen, the cell better be like something like oval, no? Better be some egg-like shape. Okay. Okay. All right. Anyway, so let's write down the note. Okay. By the end of anaphase, equal and complete set of chromosomes are found at each pole of the cell. Right. Okay. Let's see. Let's write down. So anaphase is an important phase. So write with me. Write with me, write with me. I'll use maybe purple harimokkari so that you can distinguish it from the previous phase. So this is the final phase. We are in anaphase. Okay, let's write down. Write with me. Sister chromatids. Sister chromatids are separated at the centromere. So your handwriting is smaller than mine. Okay, so you can manage the space. Sister chromatids are separated at the centromere. Then I want you to know what is the fate of the two types of microtubules. I want you to know that kinetochromicrotubules, what's going to happen to them? They shorten. Then non-kinetochromicrotubules, write down, they elongate. Non-kinetochromicrotubules elongate and kinetochromicrotubules shorten. Finally, put a star mark and you can write down. Okay, put a star mark and you can write down. Equal and complete set of chromosomes found at each pole. Equal and complete set of chromosomes found at each pole. That is the conclusion, the anaphase. So, right at the end of anaphase, right, if you watch this drama, this is what you are going to say. Come home and say, right, at the end, there was an equal and complete set of chromosomes found at each pole. So, what's next? We'll see. That is the final phase, telophase. Okay. Let me know once you are done. I don't watch too much drama. I live in an environment of too much drama. If you go to a hospital, you will not believe the amount of drama we face, particularly in female wards. Attempted suicides are there, cut injuries, paracetamol poisoning. A lot of drama is there. Okay. Yes. Most of the suicide attempts, I'm not saying they are to be taken lightheartedly, very concerning, but most of the suicide attempts are just some attention grabbing attempts of attention. So it's very concerning how people are turning to those kind of things just to get some attention. The best way to get attention is what? Do something impressive. That is a positively impressive thing, right? Positively impressive thing. That is the best thing to do, because I mean, as a girl, like so that's very inspiring. Our surgery department, our hospital surgery department is a male dominated department. We have 6 consultants, right, very seniors, and all with different personalities, very professional people are there, very eccentric people, surgeons, all surgeons have a bit of something is wrong with them. All surgeons, something is wrong with them. Either they are too friendly, or they are too strict, or they are too maybe cruelty. They are cruel towards medical students, not patients. They have a high temper. Not most of them, they have a very high temper. Those all surgeons have something wrong. So we have 6 surgeons, all different, and there's only one madam. Okay, one female surgeon, and she's like, I mean, she doesn't care that it's a male dominated ward or even department. She doesn't care. She's also comparatively young, and she's like, she has her own, many patients come just to see her. Father is a surgeon. Don't say I said all surgeons are have something wrong. Don't say that. Okay. What do I mean is they are either, I mean, with the stress of the work, they have become, they have become some eccentric people. They are all very, very intelligent people, and they are patient care. The attitude towards patients are so good. But medical students, they are the people who are subjected to the, everyone is going to blame the medical student, and you are the one who are going to get thrown out of the ward or even repeated. Okay, so you have to come on Sundays, on holidays, so that you can complete your work. Okay, anyway, right? So you will, you are, if you are, if you want to be a doctor, you are studying hard to uh, get scolded every day. So you are studying hard to get scolded every day. So keep that in mind. Okay, if you want to get into medicine, right? The only thing you should be aware of, I mean, you should be looking for in medicine is simply some satisfaction by treating a patient. That is the only thing, simply some satisfaction, not money, not mental freedom, not absolutely not there. Physical relaxation, absolutely not there. Only thing is bit of satisfaction could be there if you want to become a doctor. So many other fields are there. If you do bio, very good molecular biology courses are there in Colombo and Japura, and now in Ruhuna, Karapi, Ruhuna also there's a lot, and other sort of applied chemistry degrees are there, right? Biomedical engineering courses are arriving. So many things are there. Right? Anyway, come to the final phase, our telophase and cytokinesis. Okay, let's read the resource. Telophase. The nuclear envelope reforms around each set of chromosomes at the opposite pole. So now the chromosomes have been separated. Chromosomes have been separated. Now the only thing remaining is to build them each, right? To give them give each set of chromosomes their own cell, right? We are going to give them each of those chromosomes their own cell, right? So telophase. Highlight nuclear envelope reforms. Nuclear envelope reforms. Highlight nuclear envelope reforms. Second one, nucleoli reappears. So earlier, if you can remember, when in this G1 you had a nucleolus. This nucleolus, we got rid of it, because imagine you are about to have a party in your house. You can't have everything in your living room in there in the middle. So you are going to get rid of things that are not necessary, so that you have space. So that's what happened to the nucleolus. So nucleolus is being regenerated. So you can see, nuclear envelope is forming. Okay, nucleolus is forming. You can see here. Okay, so pay attention to these diagrams. Okay, and spindle get depolymerized. You don't need the spindle anymore. So whole entire spindle apparatus, microtubules are going to be removed. Okay, depolymerized. Microtubules unwind and become less condensed to form chromatin. Can you highlight? Chromosomes become less condensed to form chromatin. Okay, so you are unpacking the chromosome DNA, so that you have a much loose form. You can see very condensed DNA is now becoming bit longer, and they will later become thread like, strings like, uncondensed chromatin. Okay, like this. Okay, that's how a normal non-dividing cell looks like. Okay, then very important, can you highlight? At the end of telophase, two genetically identical daughter nuclei are formed. Two genetically identical daughter nuclei are formed. At the end of telophase, you don't have cells. Two cells are not there. You only still have two nuclei. Okay, we need to write down. So actually this space you have to use very carefully, because we have to write about two phases here. So the first phase, put a subtopic as telophase. Telophase. Let's write down there. Nuclear envelope reforms. Write with me. Nuclear nuclear envelope reforms. nucleoli reappears, nucleolus is singular, nucleoli is plural, because you have two, because two nucleolus are reappearing, therefore we say nucleoli reappear, spindle gets depolymerized, finally, it's not finally, okay, there's one more point, chromosomes decondense to form chromatine. Chromosomes decondense to form chromatine. Final point, put a star mark and write down. They're not final, we have something more, one more sentence to write under cytokinesis. Okay, so for now, I'm running out of space, but you can manage your space. Sorry. Two genetically identical daughter nuclei are formed. That is at the end of telophase. This is the end of telophase only. Correct? We didn't talk about cytokinesis yet, because in the same space we have to write about cytokinesis as well. I will read again. Nuclear envelope reforms, nucleoli reappear, spindle gets depolymerized, chromosomes decondense to form chromatine, star mark at the end. Two genetically identical daughter nuclei are formed. So I'm actually going through the A-level MCQ paper right now. Students started to send at this point the MCQ paper. So I need to go through it and see what would be tomorrow's fate. Okay. So the paper seems a bit easy, the MCQ. So the concern is going to be what's going to be the second paper. If the MCQ is usually easy, second paper could be a bit difficult. So, and the paper is long. The MCQ paper is kind of long. There are long questions. That's good actually, so that you can read, because in the MCQ paper there is no worry of running out of time. In the bio MCQ paper there is no worry of running out of time, never. Okay, you are going to have at least half an hour extra, because anyone, even if you didn't study, you are going to need only about 1 hour, because the thing with bio, the reality is what? If you don't know, you don't know. You can't just remember. If you can't remember, you can't just remember. If you haven't studied, you can't. Okay, but in the physics paper, if you have time, you can put this equation, you can put this equation. If you can write something, if you are doing the question again, you are getting an answer that is closer to one of the answers. If okay, you can do something with the remaining time. In the bio paper, you can't. If you don't know an answer, you don't know. But the best thing you can do is take a guess. Yes, even after taking guesses, you might have about half an hour remaining. Okay, so the bio paper, the most important thing you have to be careful about is marking the answers correctly on the your answer script. Right? The answer script with all the balls, right? Circles. That is the most important thing you have to be very careful of. Otherwise, the paper, you definitely will not have a time issue. Okay, you will have a memory issue. Okay, knowledge issue will be there, not a time issue. Okay, anyway, then we'll read cytokinesis, same page number 41. Cytokinesis. Division of cytoplasm starts at the end of telophase. At the end of mitosis, two genetically identical daughter cells are produced. Two genetically identical daughter cells are produced. Okay. So, what we are going to do is actually this. So, I need to erase this actually. This diagram, you can see there is a box in the next page of the tute. You can see this page, this box. This is actually indicating how cytokinesis happens. This is indicating how cytokinesis happens, right? So, I want you to actually on top of this, I want you to write down the topic cytokinesis. I want you to write down the topic cytokinesis. This is how actually cytokinesis occurs. Okay, this box that is A and B in a cleavage of an animal cell as well as cell plate formation in a plant cell, that is actually indicating cytokinesis. Then I want you to write down under this cytokinesis you can write produces, right, manage your space, produces two genetically identical daughter cells. Produces two genetically identical daughter cells. Okay. So, let us read about cytokinesis. Listen, come to the resource book back again, follow the tube diagram with me. In animal cells, a cleavage furrow forms. Highlight, in animal cells, a cleavage furrow forms. So, you know what a cleavage is, no? Right? You know what a cleavage is. So, this kind of looks like a cleavage, I know. Or it looks like something else also. Whatever, don't think about what it looks like. This is called the cleavage furrow, correct? So, it is formed by what? You know what is forming the cleavage furrow, no? So, who is actually doing this? Listen, what is, you can see this wrinkled parts are actually your plasma membrane, the plasma membrane, and this cleavage furrow is produced by, you can see, it is by microfilaments. You can see here around this, the place where this plasma membrane is being pulled in, it is being pulled in by actin microfilaments. So, you know microfilaments are what? You know, it's composed of actin, no? Microfilaments are composed of actin. So, there's a ring of actin that is actually pulling the plasma membrane closer together. So, what's going to happen is the plasma membranes are going to come closer, and when they connect, you are going to get two cells. That is what we call as cleavage furrow occurring in a, forming in a animal cell. So, that is only happening in an animal cell. That is only happening in an animal cell. Okay, that is only happening in an animal cell. Got it? Okay. Then what's going to happen in our plant cells? So the plant cells, you can't do something like this because it has a barrier, right? It's like your land being surrounded, the land where you live is being surrounded by a massive wall. So similar to that, there's a wall in a plant cell, no? That is the cell wall. So if you want to divide a plant cell into two, you have to build a new cell wall. Right? If your father wants to divide your main land into his two children, what does he have to do? He has to build a new wall in the center. So that's what's going to happen. Once a plant cell has been plant nuclei has been produced, what's going to happen is there will be a new cell wall being established or built in the middle of the old big cell. So you can see vesicles are forming the cell plate. These vesicles, small vesicles are going to bring cellulose, hemicellulose, pectin and other things you need to build. So when you are building a wall, what do you need? You need cement, bricks, sand, water, you need things. No? The vesicles coming to build to form the cell plate have all the cellulose, hemicellulose, pectin, those kind of things, right? Can anyone tell me who's producing these vesicles? It says here vesicles forming cell plate. Can anyone tell me who are producing these vesicles that are forming the cell plate? It's given in the resource, yes. I was yeah, I forgot that it's given in the resource right there. Okay, yeah, it's Golgi. Harry, it's Golgi apparatus. Harry, it's Golgi. Golgi apparatus is going to be producing because why? Golgi is the source of cellulose and non-cellulose components of the cell wall. Okay, so go to the next page in the resource. Highlight cell plate forms as a result of vesicles produced by Golgi apparatus. Methana, you can write on the tute. Okay, in the tute you can write vesicles forming cell plate are from Golgi apparatus. Can you write in the tute? Okay, vesicles forming cell plate are coming from where? Put an arrow. Okay, put an arrow. Vesicles forming cell plate are coming from Golgi apparatus. From Golgi apparatus. Okay. So mitosis is over, right? Let's just read significance of mitosis. Maintains genetic stability because we know it exactly copies the DNA and exactly gives it similarly divided into the two daughters. So whatever the new cell that is produced, it is not going to have any extra DNA or too little DNA. Both problems will not be there. Second, growth and development, of course. If you damage one of your skin epithelial cells, right? If you make a cut here, all the skin epithelium as well as some tissues are there that is called the connective tissue, they are going to divide by mitosis, and then there will be the wound will be repaired. Okay. Then cell repair. If you damage a certain cell, it will get replaced by a new cell produced by mitosis. Replacement and regeneration. Yes. So if there is a certain cell that got damaged, particularly it mostly happens in your epithelia. That means in your inner surface. That means From your mouth to the anus, there's your gastrointestinal system. On your surface, you have your epithelial skin surface. Those kind of things, those kind of cells rapidly get regenerated. For example, in your stomach, it gets commonly damaged with acids. But rapid mitosis is going to regenerate more and more cells faster than they die. Faster than the cells are dying of the acid HCl in the stomach, more and more cells are being produced, so that you don't get what? According to, okay, is it a signal process? Wound grow faster. There are things happening when you have a wound. Okay, when you get a cut or something, there is something called as an inflammatory response happening. It's a part of the immune system. So when a cell is damaged, it's going to release certain chemicals that are going to it is going to ask immune cells to come there, because why? When you have a damaged area, it's a good place for bacteria, pathogens to enter. So it's going to need some defense. So white blood cells are going to come. At the same time, more proteins are going to come there, so that whatever the cells that are surviving, they are able to divide and somehow replace the damaged cells, so that you have, mostly you will repair your surface. Okay. Asexual reproduction, that is the other thing. Animals such as, hydra, certain cnidarians, jellyfish mainly, those kind of animals, they are able to give rise to an exact organism like the parent, right? Exact. So they reproduce by mitosis. Okay, so they don't need two people. Then we spend a lot of energy finding another partner. No? So organisms who reproduce asexually, they don't need another partner. They will do it by themselves. They will give rise to a new young organism by themselves. Single parents. Okay, asexual reproduction. Clear? Okay. There's a small label here. What did we? Yes. Can you label here? We couldn't label this one. This box. This is what? This is cleavage furrow. This is cleavage furrow. Okay. So mitosis is over. Now I want you to go into in the resource book. We are going to skip some pages. We are going to skip meiosis, and I want you to come to tumor, cancer, and goals. That is page number 47. Tumor, cancer, and goals. Page number 47. Okay, because you mainly, because why I am doing this right now together with mitosis, it's a problem with mitosis usually. Mitotic division is the cell division that is usually causing most of the cancers, right? It's not most of the cancers, almost all cancers are formed usually due to errors in the cell division, right? And so, like, let's say the error could be in the cell division process, or it could be in the cell itself, so that the cell got abnormal, and then when it's dividing, it is dividing abnormally. Okay, so I have given here what's the, let's say, the progress, how a cancer originates, right? Every cancer starts with a single cell, right? The example I like to use is a criminal organization, a massive criminal organization, Al-Qaeda, would have been started with one man, with the idea to start a criminal organization is enough. Okay, so the cancer that is going to spread, this sort of big cancer, is going to start with a cell with a mutation. A single cell is going to be the person starting this. Okay, can everyone come to page number 47? Okay, page number 47 in the resource. Let's read. Okay, Cell division is driven by external and internal factors. They may be chemical or physical factors. Can you highlight? There are external and internal factors. External means outside the organism's body. Okay, outside the organism's body, such as the things that he is exposed to. He might be getting exposed to what? Certain viruses, certain radiations, X-rays, gamma rays, alpha rays, right? Then what else? He could be drinking some water containing heavy metals, or he's being, he's consuming alcohol, he's smoking, right? All those things are external factors. Internal factors are mostly genetical factors, okay? Then, so that's what they say, they may be chemical or physical factors. So, if you take the alcohol and the cigarette smoke, they are chemical factors. Then if you take things like being exposed to X-rays and UV rays, those are like physical factors. So, okay. Cancer cells do not respond normally to the body's control mechanism. Can you highlight? Cancer cells do not respond normally to the body's normal control mechanism. They divide excessively and invade other tissues. If unchecked, they can kill the organism. Okay, if unchecked, they can kill the organism, because why? You will see the new, so a cancer starts with one cell, that cell will start to divide, and when it's dividing, because it's an abnormal growth of cells, these abnormal cells are dividing too fast, that's the point. They are dividing too fast, therefore they are not able to sustain themselves, they can't even keep themselves alive. So when the cancer is dividing, some cells are going to die. At the same time, because of the excessive division, whatever, so imagine you have a cancer in your lung, the new cells that are being produced are not lung cells, normal lung cells, never. That's being produced. The cells that are being produced are abnormal, useless cells. So you are going to lose function of your lungs over time. So the person with time, the person is not able to extract oxygen. So the person is going to die. Okay. So they divide excessively and invade other tissues. If unchecked, they can kill the organism. Can you highlight? They divide excessively and invade. The main thing about a cancer is this invasion. Okay, page number 47. Right, cancer cells do not consider. So we'll read and we'll make a note. Okay, I'll explain show you some images as well. Cancer cells do not consider the normal signals that regulate the cell cycle. Okay, so you know there are cell cycle checkpoints, no? Yeah, those checkpoint signals, they don't care. They do not need also growth factors. So growth factors could be certain nutrients, or it could be certain hormones. For example, testosterone mainly sort of makes the skeletal muscle growth. Those kind of hormonal signals are there. Okay, so those are called growth factors. So those kind of growth factors are not required for cancer cells to divide. Okay, they make the required growth factors themselves and give signals to continue cell cycle without growth factors. Yes, there are growth factors such as we call as tumor necrosis factor. Those are growth factors that are, growth factors are like chemical signals that are produced by the cancer itself. So he is going to produce signals for himself and he is going to divide. It's very sustainable level, very independent things, cancers. That's why they kill. Is it possible to find cancer on the first stage? Certain cancers you can find early, but some cancers, until you get severe symptoms, you will not identify. Mostly lung cancers are very silent. Lung cancers are usually silent. The person usually only has maybe a cough for a long time. Now and then he has some difficulty breathing. Okay. Then he thinks, okay, it's a small pneumonia, hari cold or something, cough, no, right? So he's just going to ignore until the lung is entirely invaded by cancer. Then there are very invasive cancers such as ovarian cancers. Females get in their ovaries, nothing, no symptoms. So what we mainly train, so I had 2 months in Apeksha hospital. What we were asked to look into in our patients by our consultant was, how are they coming to the hospital? The person is now in the ward with ovarian cancer. So what our consultant wanted us to know mostly was, why did they come to the hospital in the first place? So that was why they got diagnosed with a cancer. No? So they want to know why did they come? So if you learn how a certain cancer presents, what are the first signs of a cancer, you can stop the cancer in the first stage, right? Certain cancers such as ovarian cancer, nothing. The person doesn't feel anything. Sometimes the ovaries are massive, they are filled with cancer, but the person only may be having a small abdominal pain because you don't feel anything. Okay, randomly if someone had an ultrasound scan in the abdomen, randomly they will find out, okay, massive ovarian cancer is there. So it's by chance. So some people get checkups done, they get MRI scans almost every year. Okay, that's good. They can identify cancers, some cancers lung, and lung cancers maybe, and certain prostate cancers can be identified by MRI scans. Some people get checkups done, that's good. If you can afford, that's good. Okay. 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 They possess an abnormal cell cycle control system. Can you highlight? They possess an abnormal cell cycle control system. So everything about them is abnormal. The problem begins with when a single cell undergoes transformation. Meaning the single cell with a mutation, right? The man who's going to start everything, and this process converts a normal cell into an abnormal cell. If the body's immune system cannot recognize and destroy it, yes. If so, for my 27 people, I just finished my the immune system. There's a special type of immune cell that is called natural killer cell. Right? Natural killer cell. So as the name says, he is a very dangerous cell. He is able to kill other cells that he thinks are not normal. He is able to kill any abnormal cell. Usually, he is the main defense in your body against cancers. Natural killer cells. Just keep in mind that I told you this right now. When you are doing immunity, at least you will think of what I told you. Natural killer cells are your main cancer defense. Okay. It may lead to proliferation of cells and formation of a tumor. So you have a ball of cells. If the abnormal cells remain at the original site, the lump is called a benign tumor. Can you highlight? That's a definition of what's a benign tumor. The words you need to know very carefully are what is benign and malignant. Okay. So what we are going to do is we'll make a small note comparing the two types of cancers. Okay. All right, we'll make a small note. Okay, so you can come over here and we can start writing a small note. Okay, first point, tumors originate when a single cell in a tissue, gets transformed to an abnormal cell. That is how the cancer starts. Next point, if it escapes the immune system, it will proliferate and become a tumor. Then what we are going to do? So where are we right now? We have one abnormal cell. That cell started to divide. That's what we mean by proliferate. That means rapidly dividing. Now you have a lump of cells that we call as a tumor. Tumors can behave in two ways. That there are dangerous tumors, then there are let's say a bit less dangerous, or you sometimes even harmless tumors. So you can, I want you to classify two types of tumors. One is benign, the other one is malignant. Okay, write this down quickly. There's this, I'll send this, there's a comedy sketch from this old British TV show, right? I think I can just show it to you. I don't know if you can hear it, okay?
We've had your test results back, and you'll be pleased to hear that it's benign.
Oh, thank goodness. That is such a relief.
Yeah, it's good news, isn't it, Mr. Ignant?
I'm not Mr. Ignant. I'm Mr. Ein. Mr. Ben Ein.
Well, who's this Mal Ignant? Well, we've had your test results back.
Right? Did you get it? Right? So we'll see. I'll teach you about malignant tumors, then I'll show it to you again. Then hope you can understand. Okay. So quickly write this down. Benign versus malignant. There are two types of tumors. Okay. So malignant. So before we move on to malignant, the nature of a benign tumor, is that it doesn't go anywhere. It doesn't exactly feel like moving a lot, right? It doesn't feel like, the word is invading. Benign tumors do not invade. That means go into other locations. So we can write down on the benign side like this. Remain at the original site. Remain at the original site. Next point, most do not cause serious problems. Most do not cause serious problems. Remain at the original site. Most do not cause serious problems. Next, can be surgically removed. You can usually remove a benign tumor surgically. Can be surgically removed. By doing surgery, you can remove. I'll show you some images. Can be surgically removed. Then you come to the dangerous type of tumors. Because of their inherent sort of nature, the word we use to describe them is they are invasive. They are invasive. So let's write down on the other side, malignant side, they are invasive. That means they invade, they forcibly go into other tissues. Write down under this, travel to other parts via blood or lymph. Travel to other parts via blood or lymph. Then what they do is, then they attack other organs. So imagine our country was invaded by another enemy foreign country. What they are going to do? Once they are here, they are going to destroy everything. They are going to take all our money and our resources, right? So that's what's going to do. When the invasive malignant cancer is at a certain place, at a new place, he is going to extract all the information and he is going to kill whatever that is around there. That's how a malignant cancer works. Attack other organs. Okay, attack other ones. I'll erase this. Okay, make on man. Attack other organs. Then we have a special name for a person who's having a malignant tumor. We'll say here with a different color pen, if you can write down better, a person with a malignant tumor is said to have what? A cancer patient. Okay, you are diagnosed with a cancer. Then we talked about something called as travel to other paths via blood or lymph. That means these malignant cancer cells, they are very traveling. I mean, so they are like a traveling, foreign traveling enemies. Okay, so they have several types of traveling highways that they like to use. If you zoom into this diagram, imagine you can say there's a primary cancer here in the large intestine here. So it can enter into your bloodstream. Can you see? It's entering into your bloodstream and traveling via blood into distant organs. Or it can enter into there's a system called the lymphatic system. You might have heard the lymphatic system is like our backup drainage system, right? It's our backup drainage system. So the lymphatic system is also part of the circulatory system. So the cancer cells can also spread to the lymphatic system and also spread through there. So you can see cancer in the large intestine can spread to the liver, can spread to the lungs. Okay, so that spreading of a malignant cancer into further distant locations has a nice name. It's called, not nice, sorry, it's called metastasis. Okay, I will zoom out and write. Okay, so you can put a small subtopic here, metastasis. What do you mean by metastasis? Okay, let's write. The metastasis means spread of cancer cells, to locations distant from their original site. Spread of cancer cells to locations distant from their original site. So here in this example diagram, the cancer started in the large intestine, but it has spread to, usually, right, liver is a very common place of cancer metastasis. Right? Because why? The liver gets a lot of good nutrients. No? All the nutrients from your body goes to the liver. Liver has a very good blood supply as well. Then the lung is also a favorite place of cancer cells to go, because why? It gets a lot of oxygen as well as a lot of blood. Your lungs very rich in good blood supply. Okay, so they like to grow there, where you get a lot of nutrients, oxygen, blood, very good places to grow. Okay, so that is called metastasis. Then I want you to draw two arrows. Metastasis is of two types, as blood metastasis and lymph metastasis. There is blood metastasis as well as lymph metastasis. Okay, blood metastasis as well as lymphatic metastasis. Quickly finish writing this. I will zoom out so that you can complete the note. Okay, you can see, no, malignant tumors are invasive. They travel to other parts via blood or lymph and attack other organs. Then special point, a person with a malignant tumor is said to have a cancer. Okay. Tumors and cancers are not the same. That is, again, you are right, and as well as not exactly right. tumors are of two types. There are invasive tumors as well as non-invasive tumors. Okay. So a person with an invasive tumor is having a cancer. So it's like it's a common area, right? Tumor and a cancer is commonly overlapping. If the tumor is an invasive tumor, then it's a cancer. Right? So the tumor is the big term. Within tumors, you have cancers. If you really want a tumor, what do you want? What type of tumor do you want? You want a benign tumor. If you really need to have a tumor, you better have a benign tumor. But what's the bad type of tumor to have? Malignant. Don't have a malignant tumor. You don't have a choice, right? Which sentence? Second sentence here under tumor, cancer, and goals. If it escapes the immune system, it will proliferate and become a tumor. If it escapes the immune system, it will proliferate and become a tumor. Okay. Right. Then I can. Yeah. I'll show you. Okay. There are things called as local effects as well as systemic effects of a cancer. Okay. Of a tumor, there can be local effects. I'll show you. Okay. So, okay. Are you done writing? Can I show you some images so that all these doubts will be cleared? Okay, I'll go back. Okay, so now I already started. I'll show the written things again. Okay, right. So this is something what I want to tell you about a benign tumor. Right, benign tumors are like this. Benign tumor, if you take a breast, right, the benign tumor is just a very nice ball of cells. It has nice margins like this. It's simply an abnormal lump. Okay, in the breast or something, right? It usually doesn't try to spread anywhere. It's going to get bigger, so that you feel like a big lump. If it occurs in the uterus, it's just a big lump of cells that are sort of making you can feel it. It's very heavy, and also it has what we call as pressure effects. It can compress some other things in the surrounding area, causing some symptoms. Okay. So this is the problem with a malignant tumor. Right? You can see this is the malignant tumor white color parts. Right? You can see it is not like a very nice spherical ball of cells. No, it is spreading everywhere. Can you see it is spreading? This is all fat cells in the breast. This is a breast cancer. You can see this fat yellow color is the fat cells in the breast. The white color is the abnormal cancer cells that are spreading everywhere. So we say the way we explain them is they are invasive. Okay, they are invasive. Then you can see here a nice example of a benign versus a malignant. A benign cancer tumor is going to get bigger until you have a big ball of cells. So, by surgery, you can open up the uterus, you can take this ball of cells out, and everything will be okay. Usually, we do this if the tumor is too big. Otherwise, we don't even sometimes do surgery because it's just going to be uh, unnecessary surgery. Okay, but here you can see this is a malignant. You can see it doesn't have a definite shape. It's just some cells that are dividing. At the same time, they are invading. So in the invading places, you can see there are cells, I mean, there are dead areas. You can see there is bleeding. That's because cells are dying. Your blood vessels are being damaged. Okay, your blood vessels are being damaged. So this is not a very good type of tumor to have. Malignant. Okay, this is also a liver uh, malignancy. You can see it is dying. I mean, the cells are dying, therefore there is bleeding, and the cancer doesn't have a definite shape. No, it's like spreading everywhere in the liver. Okay, this is also a breast malignancy. You can see this is the nipple of the breast. It has been pulled in because all these cancer cells are invading everywhere and damaging the breast. Okay. What else? So that's you don't need the microscopic differences. And what else can I show you? Yes, certain conditions. That means is origin of a cancer in the esophagus. That means the food pipe. Okay, lower part of the food pipe near the stomach. You can get a cancer like this. So accidentally, if you put a camera that we call as an endoscope, I really enjoy endoscopic views. No, I have shown you several, right? Accidentally, if you put a camera for maybe to treat gastritis or something, mockery, then you notice this kind of abnormal appearance. That could be your luck, so that you have diagnosed early esophageal cancer, right? Early esophageal cancer can be diagnosed like that. So this is how metastasis occurs. This is another diagram showing how different metastasis can work. Okay, if you have a primary breast cancer, it can spread to the brain, to your bones, to the liver. Okay, so many places it can metastasize to. That's why the person dies. A person with breast cancer, that's why they die, because it has metastasized to important organs, and the important organs are shutting down. They are being destroyed by the abnormal cells. Okay. Yes, you can see some examples. You can see this normal, this sort of red, dark red colors areas are the normal liver cells. These white lumps are abnormal cancer cells, so they are useless. So the liver can't do its normal function, so that the person doesn't have a liver actually. So that's why they die. This is a lung. Okay, so this normal sort of brown color area is the normal lung. You can see so much white patches. Those are liver lung tumors that are spread here. Oh, it could be primary. It could have originated from the lung also. No, cancer can originate from the lung. This is bone, right? This is a bone infected. I mean, sort of, there are so much cancer cells here. One of the biggest problems cancer patients have in the later stages is they get bone pain. Right? Bone pain because of the bone metastasis. They get a lot of pain. Even strongest painkillers can't reduce that pain. Very severe pain. Okay, and this is in the vertebral column. In the vertebral column, you can see your back. There are so much cancer cells in the vertebral bones. Okay, so that's basically it. What I need to show you. Yeah, it looks like fungus infected. Yeah, it looks like fungus infected. Yes. Okay, quickly complete this. Anything what's missing? Can you go down? Okay, Mithana. Okay. How many ways there are to get cancer? So many, right? Mostly it's acquired, things because of people's habits. Usually genetic predisposition is not much. Lung cancer, it's cigarette smoke, also asbestos, arsenic, certain silica dust, oral cancer, beetle chewing, bulat. Yes, being in the sun is dangerous. UV rays. Without sunscreen, don't go to the sun. You get a skin cancer. Have I watched Grey's Anatomy? Not much, because I find it a bit not very realistic. I mean, most of the medical dramas are not realistic. Foods that reduce cancer. If you want to reduce the likelihood of oral cancer, your gastrointestinal, usually lower intestine, that means colon, rectum, eat a lot of fiber containing food. Fiber. right? Fiber containing food mostly, and try to reduce the red meat, mostly beef. Right? We don't eat a lot of beef, no. So in Sri Lanka, colon lower intestinal cancers are not very common. In Sri Lanka, the commonest cancer is breast cancer. Actually, it's the most common cancer in the world, breast cancer. Then the commonest cancer is mostly lung cancers. In Sri Lanka, not lung cancer, second place. It is skin. Anyone know what is the second commonest cancer in Sri Lanka? Oral, mouth, because of the betel chewing as well as the cigarette smoking. Right. Eating meat with cancer will not give you cancer. Okay, they will be broken down by your digestive enzymes. To get a cancer, your own cells should be abnormal. To become, they will should become abnormal because of either what, chemicals or physical agents. Your own cells should become abnormal by eating certain, a person with cancer cells. You will not get a cancer. Okay, they will be broken down with your digestive enzymes. Anyway, okay, we have another small part to be completed. That is how plants get cancer, plus certain abnormal cell growths in plants. So with that, we are done with tumor, cancer, and goals. Does sunscreen cause cancer? No, they protect you from skin cancer. Wear sunscreen. Okay, don't go outside without wearing sunscreen. Okay. Then can you come to page number 48, goals in plants? Can you come to page number 48? Goals in plants. Okay, let's read and highlight. This occurs due to uncontrolled mitotic division of a plant cell. Can you highlight? Uncontrolled mitotic division of a plant cell causes goals. The plant cell division is controlled by maintaining a proper balance between plant growth regulators such as auxins and cytokines. So similar to the hormones that you have in your body, plants have also hormones. We actually call them not by hormones, we call them as plant growth regulators. Can you highlight? Plant cell division is maintained by a proper, controlled by maintaining a proper balance between plant growth regulators such as auxin and cytokines. You will learn about them in detail when we are doing fourth lesson, plant form and function. Okay. When this balance is lost, plant cells produce undifferentiated mass of cells. So in plants, whether to divide or whether to stop dividing is controlled by these two types of plant growth regulators, which are actually chemicals. So usually auxin stop plants from dividing, plant cells from dividing. Cytokinins usually stimulate them to divide. So always you should have, like a seesaw, a balance between auxin and cytokine. Otherwise, there could be abnormal cell growth in the plant. Okay, then there's a definition. I want you to take out your definition color, highlight a color. Goals are bumps and growths that develop on different parts of plants after being invaded by some unique organism. That's a definition. Okay, let's write here the same definition. I want you to, can you come here on top of this diagram? Can you put the topic? This is a goal, Hari. You might have seen, no? When you are walking outside on trees, on the stems of trees, you might have seen this sort of abnormal looking black or dark colored lumps on the plant body. That's it. The plant is having a tumor. Okay. Write down the topic, goals in plants. And then let's write down the definition of a goal. Okay. Goals are bumps or growths. Goals are bumps or growths that develop on different parts of plants after being invaded by some unique organisms. Then you can write down what are the organisms that can cause. Usually listen, a plant is getting usually these galls or abnormal masses or bumps of cells due to an attack by another organism, by due to an attack by another organism. So I want you to remember what are the attacking organisms. Usually there could be viral based, okay, viruses, bacteria, very common, right? In your syllabus you have a bacteria that is causing a bump. We talk about it in microbiology actually. Right? Bacteria can cause insect attacks, because once a certain insect attacks, due to, because the damage caused by that insect, the plant actually starts to produce more cells, and then those cells become abnormal because the plant is sort of like overreacting. The plant is overreacting to the attack, so that those additional cells become cancer cells. Sorry, I mean sort of abnormal cells. Insects and also mites. So mites are also, I think, insects tamai. Okay, viruses, bacteria, fungi, no, mites and fungi. Write down. Galls are bumps or growths that develop from different parts of plants, which are after being invaded by some unique organisms such as viruses, bacteria, insects, mites, and fungi. Okay. Okay. All right. Done. Right. So what we can do right now is we can take a small break, and then after the break we'll finish meiosis. Okay. Right. Then shall we take a small break? Come back at 8. Okay. Break. Okay. Rejoin at 8. Take a small break and quickly rejoin, where we'll be finishing meiosis. Okay. If you have any questions, send them to the chat. Are there brain cancers? Usually brain primary origin is not usually there. Brain, usually cancer spread to the brain. Okay. Primary origin is not usually there. Usually cancer spread to the brain. Okay. All right. Take a small break. We'll rejoin at 8 p.m. Okay. Okay, right, let's continue. Okay, come back. We have to go back to, we have to finish meiosis. Okay, all right. I was having a chat with the 26 people. Okay, so they have they came home at around, the paper finished at 3, they all came at around home at around 4. So the tomorrow's paper is going to be at 1. So they have around less than 10 hours. So 10 hours means they have to sleep at least 6 hours. Okay, so no, sorry, they have 24 hours. Okay, so they have to sleep at least 6 hours. So they have maybe around 12 hours proper to work with the meal time, traveling and getting ready, all the things when they have removed that. So okay, so at the end, all of this 2 years of learning is going to come down to that. Okay, so keep that in mind. So there's going to be a limit of what you can cover within those 12 hours. But if you have everything retrieved in your memory, if you have them nicely organized on the 12 hours before the second paper of your exam, you are going to be able to take them all out very nicely. Okay, that's my goal, so that you have from the beginning to the end of the resource book, you have a nice organized packaging in your brain of all the theory points. Okay, otherwise that is the best way, that you don't panic the day before the exam, because you can see all the completed tudes that we have done, all the completed questions tudes we have done, and once the paper class has started, you can see all the completed papers that you have done. That is going to give you the sort of the strength as well as the confidence to go to the exam, man. Because at the end you know everything, and you have studied, it's in your memory. What's going to be your biggest enemy is you getting nervous, getting anxious. That is going to be the biggest enemy. So you have to keep your mind at on the goal. Otherwise, it is going to be a difficult time, this final few hours before the paper. But if you have trained to that mentality, if I'm keep on telling you to, because I want to get you what sort of a mentality it's going to be in those final hours. Okay, so that's why it's not something that's all. So I hope it won't surprise you how anxious you can be the few hours before the exam. Okay, so that's why I'm keep on saying you are going to face this sort of anxiety before the paper. So I'm trying to tell you, you have to train yourself from right now to stay cool, stay calm before the paper. Okay, I'm saying this from my experience. Okay, as a teacher as well as a A-level student, I'm saying how my students react the day before the exam, as well as how I felt the day before the exam. Both are adding up to my experience. That's what I'm telling you, because mostly what I want to tell you is, as a student, right? Because I was in the middle now, I am sort of gathering my experience as a teacher, as well as I can still remember how I went through this as a student. So that's the thing, right? So as a person in my age, I am having that sort of balance. So I remember how I felt as a student, and now as a teacher with several batches, now I know how you guys feel as well. So that's what I'm trying to do. No matter how much you learn theory, how much I teach you theory, the way you feel the day before the exam and at least the few weeks before the exam, is going to be the main determining factor. So we are working and collecting notes. We are doing so many elaborate things so that you feel confident before you go and face the exam. Correct? That's what I want to tell you. Anyway, okay, let's come to the resource. Let's go into meiosis. Okay. Page number 43, meiosis. Page number 43, meiosis. It's not that complicated, right? I don't want you to become let's say, don't worry about meiosis a lot. If you start If you think about it lightly, it will come to you. The theory, you will be able to understand without a doubt. Okay. So don't worry about it too much. Just follow me with whatever is in the resource and I'll explain. Then we'll complete the tube. That will be more than enough. Okay. So don't overthink. That's the main thing I want you to not do. Okay. Let's see. Meiosis. Sexually reproducing organisms undergo different type of cell division called meiosis. Can you highlight sexually reproducing organisms? Meiosis is a type of nuclear division which gives rise to 4 haploid, genetically non-identical daughter nuclei from a diploid mother nucleus. Can you highlight? That is the definition of meiosis. It's a nuclear division. Nuclear division is very important. Mitosis or meiosis, both is a nuclear division giving rise to 4 haploid. I will explain what is haploid. Genetically non-identical. Haploid is very important. Genetically non-identical. So there's a lot of differences in meiosis compared to mitosis. How many? They are haploid, they are genetically non-identical, as well as there's going to be 4, not 2, similar to mitosis, from a diploid mother nucleus. Okay. So let's see. Okay. Yeah, let's keep on reading. You can see here this summary, no? So you have a diploid cell. That means, what do you mean by diploid cell? So listen, you can take this also, but this is a better diagram. Okay, now listen. You can see in this organism we have used now red and blue color. We have used now red and blue color. That is because you are a product of your mother and father. You are a product of your mother and father, right? So DNA from your mother as well as DNA from your father is present in you 50-50, exactly half and half, right? So if you take your human body cells, you have how many chromosomes in total? You have 46, no? You have 46 chromosomes. Out of them, you got 23. I'll use the red color here onwards for mother's DNA chromosomes. You got 23 chromosomes from mother and 23 from your blue color. I'm going to be using for father. 23 from your father. That's how you got your 46. For simplicity's sake, we are going to study cells with less number of chromosomes. We can't draw 46 in all the examples. So for simplicity's sake, we are going to use chromosomes with less numbers. Let me check. Okay, yeah. We are using, can you see? We are going to be using a cell here. Wait, before showing you this cell, I will go back to this, yes. Okay. So you can see this cell that we are going to subject to meiosis. You can see how many blue chromosomes can you see? 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 Here, this is the original cell before it has undergone our S phase. Okay, because before it has undergone S phase, this is the cell. So it has given, you have been given a number 2n is equal to 6. So if you take your cell, your 2n number is equal to 46. No? So for simplicity, we are considering a cell only having how much? 6 chromosomes. So if you have 6 now, you can see you got 3 from your father, 3 blue, and 3 from your mother. So altogether 6. Right? So that is the simple cell that we are going to consider in our examples. Right? Then with that understanding, okay, so now this is the original mother cell, right? This is the parent cell. Before it is going to become, its DNA is going to become doubled. So you know, before even mitosis, so if you think about mitosis, all this DNA is going to be doubled, no? Because all the chromosomes are going to have a sister, no? So this blue one is going to have another sister, no? This blue one is going to have another sister. It's like when there are originally blue one chromosome, now he is going to get an exact copy. He is going to get an exact copy, no? That is what we mean by getting sister chromatids. That's what's happened in chromosome duplication. We call it what? DNA replication which occurs in the S phase. Okay, so this is what it looks like after the DNA of each of the chromosome got doubled, right? So you know in mitosis, then what's going to happen? These are all chromosomes. These are all chromosomes with two sister chromatids, no? Two sister chromatids. So what's going to happen? The sister chromatids are going to be separated. You are going to still have two daughter cells with same ploidy, no? So you are going to have 2n is equal to 6. You can count, no? 1, 2, 3, 4, 5, 6 cells. Chromosomes 1, 2, 3, 4, 5, 6 chromosomes. So 2n is equal to 6 is going to be maintained at the end of mitosis. Meiosis is a whole different game. Alright. So now listen. You started with a diploid cell 2n is equal to 6. Now jump to the end of the daughter cells where you have the daughter cells. You can see they are all n. No? N means what? How many chromosomes can you see? 1, 2, only 3. So n is equal to 3, n is equal to 3, n is equal to 3, n is equal to 3. Your daughters are haploid, half the number of chromosomes. The other thing is what? You can see all the daughters are completely unidentical. If you take a look at the daughters of mitosis, you can see there are 3 pure blue cells, well, there are 3 pure blue chromosomes, and then 3 pure red chromosomes here. Then on this side, there are 3 pure reds and 3 pure blues. But what has happened here? You can see, if you take this daughter cell, there is a pure red one, there's a red one with a piece of blue, there's a blue one with a piece of red. If you go to this one, there's a big red one with a big piece of blue, there's a small red one, there's a small blue one. So you can see these two guys are unidentical, no? These two cells are unidentical. Then if you go here, there's a big blue one, there's a blue one with a red one, there's a red one with a blue one. So each of them are unidentical to each other. That's how the daughter cells of meiosis are like. Okay, that you have to keep in mind. And the other thing I want you to remember is meiosis is different from mitosis by the number of phases you have. Meiosis has 2 phases of division. That is, this whole thing is called the first part of meiosis. It is the meiosis 1. Meiosis 1 has individual what? Meiosis 1 has individual, I will just going to say prop phase 1 is going to be there. There's going to be another metaphase 1. There's going to be a anaphase 1. Then there's going to be a telophase 1. And there's going to be a cytokinesis. This is all for meiosis 1. Then you have meiosis 2 also. There's another part of meiosis. It is also going to have a prophase 1. It is going to have a metaphase 2. Sorry, I'm sorry, 2. 2. Prophase 2, metaphase 2, anaphase 2, telophase 2, and there's going to be another cytokinesis. Okay, so the meiosis is a bit longer as well as a bit more complicated because of all these differences. Clear? And so now with that knowledge, we can move on to what are the individual things that are happening in each of the phases. Okay, so let's keep reading. Meiosis involves 2 consecutive nuclear divisions. We are now in the resource book. Okay, meiosis 1 and meiosis 2. Very important statement is there. Meiosis 1 is a reduction division. Meiosis 2 is similar to mitosis. Can you highlight? Very commonly questioned in MCQs. Meiosis 1 is reduction. Meiosis 2 is similar to mitosis. We say that, now I will explain, but with time you will again understand. So up to here I told you this is meiosis 1. No? Up to here this is meiosis 1. You can see when you are starting meiosis 1, yes, when you are starting meiosis 1, how many chromosomes can you count? How many chromosomes can you count? Yes, you can, if you zoom in here, you can count 3 red, no? Why are they doubled? Because they underwent DNA replication, no? So you can see there are 1, 2, 3 reds. Yes. Then you can see 1, 2, 3 blues. Yes. What's going to happen is, so they are going to undergo metaphase 1. Then there is something called anaphase 1. Now can you see how many chromosomes are there at the end of meiosis 1? There's only 1, 2, 3 chromosomes. I know they are doubled, but if you take them together, that's a chromosome. No? So there's only 3 chromosomes at the end of meiosis. So at the end of meiosis 1. So meiosis 1 is going to do the conversion of diploid to haploid. Meiosis 1 is going to do the conversion of diploid to haploid. Right? Then I'm just giving you an overview. We are going to go each of the steps step by step. Okay. Then you move on to meiosis 2. In meiosis 2, what's simply going to happen is, similar to mitosis, in the middle of this red one, it's going to be separate. In the middle of this red one, it's going to be separate. The sisters are going to be separated in meiosis 2, similar to mitosis. Sisters are going to be separated in meiosis 2, similar to what happened in mitosis. You can zoom out and you can see here, no? Because the sisters are being separated in mitosis. No? In meiosis 2, sisters are going to get separated so that you have a division that is similar to mitosis. You have a division that is similar to mitosis, which is meiosis 2. Okay. Clear? Okay, so with that knowledge, let's move on to a concept that I want to explain, that is called homologous chromosomes. The next concept I want to explain is called homologous chromosomes. So I want you to write down a small note here. Let's write with me while I am explaining. Okay, while I am explaining, let's write. Can you come to this page of the tute? Okay, that is page number 11. Okay, come to page number 11 of the tute. Let's write down a topic called homologous chromosomes. Homologous chromosomes. Right? Let's write down. Write with me. Okay, I'll explain after we have written. Okay. A cell having two sets of chromosomes, one set from the father, within brackets අපි එකට කියනවා paternal, one set from the father, paternal, and one set from the mother, we call that maternal. That cell is referred to as diploid. That cell is referred to as diploid. Within brackets, write down the symbol of diploidy, 2n. Alright. Next point, let's write down. Okay. I'll explain. In a diploid genome means the set of all chromosomes. That means if you extract all the DNA in a cell, that is called as a genome, the collection of DNA. In a diploid genome, maternal and paternal chromosomes can be paired up as homologous pairs. Can be paired up as homologous pairs. Okay. Next point is an important point. Put a star mark. Homologous pairs are similar in these features. Homologous pairs are similar in these features. Let's write down. So that's all you need to write down here. There's going to be a small diagram that you need to draw. So keep some space there. Okay. They are similar in shape. Write down the things that are similar among homologous pairs. Similar in shape, length, 3, location of centromere, and finally sequence of genes. Sequence of genes. Homologous pairs are similar in these features that are their shape, length, location of centromere, and finally the sequence of genes. Okay. Okay. Any questions? Yes. This is more diagram to be drawn here, but I'll draw it in my whiteboard. So whiteboard means, so keep some space to draw a small diagram in this space. Okay. So don't write big letters like me. Okay. I know you have some good space there. Okay. Okay. Right. Anyone who's still writing? No? Okay. Oh yeah, so this is what I used to explain last week. What do you mean by kinetochore? Okay, then I think I explained to you what do you mean, what is how DNA is packaged also, how chromatin is formed, and how chromatin is going to become chromosomes. All those things I explained. So today I want to explain a concept that is called the homologous chromosome. So you know, if you take your chromosomes after they are formed, I mean after chromatin becomes chromosomes, You are going to have chromosomes numbers from chromosome number 1, 2, 3, 4, 5, 6 so up to 46 when you are going to have chromosomes, no? Right? So actually you are going to not have entirely 46, but we say you have 23 pairs. 23 pairs you have. That is because imagine you got one chromosome from your mother, we'll call it chromosome number 1. There's going to be a chromosome you received from your father that is similar to that mother's chromosome in what? Shape, what else? Length, centromere location and sequence of genes. Alright. So, because why? Your father was a human, your mother was also a human, no? So the half of DNA that your father is giving should be equal in these features. Hurry, be very careful. They are not the same information, but when they become chromosomes, they have similar shape, similar length, similar centromere location, those things. They are not having the same information because your father and mother were not twins, no? Okay, so they are not having the same information, but they are similar in their in those features. Okay, right? So because I mean similar to this mother's first chromosome that you got, there's going to be a chromosome from the father. That is going to be your first chromosome pair. That is going to be your first chromosome pair. There's going to be a second chromosome that you got from your mother. We'll draw a very thin and long one. We have thin. There's going to be a similar chromosome that you got from your father that is having the same shape, length, centromere location, all those things. Then let's say there's going to be a third chromosome that you got from your mother, fat and short toiga, that is going to be have a similar shape, length, centromere location chromosome you got from your father. Hurry. Now the thing is, listen, the genes of these chromosomes are not the same. What do you mean by genes? They have sequences of DNA that are determining certain characters, your skin color, hair type, eye color, those things. Right? So in the same places, you are going to have the same type of gene. You are going to learn about this in DNA, genetics. Okay. So imagine here in your mother's chromosome, you have the gene for curly hair. In your father's chromosome, in the same place, you are going to have a gene that is determining the hair type. But it doesn't have to be the same gene. So your father might be giving you straight hair. Father's hair determining gene could be a gene of straight hair. Mother gave you curly hair. Father is going to give you straight hair. So what type of hair you are going to have is going to determine later on other things, such as how those genes interact. Okay, you don't need to know about that right now. Okay, so this is what you call as homologous chromosome. We call this, right, when such a maternal and paternal pair you call as a homologous pair. That is the knowledge I want you to have. Right. Can you draw this? There in the diagram tute where we wrote down the note. Can you write this? So you can label them. This is the maternal chromosome. The blue one, you can label it as paternal chromosome. Okay, so there in that space you don't have to write anything about it, anything else. Okay. Yes, the genotype is something you have in your genes. The phenotype is what you can see from your outside. So the genotype is going to determine your phenotype with certain other interactions. Could be the environment, Okay, right. So with that understanding, I want to just no need to write anything down. Okay. So now you have an understanding what do you mean by a homologous pair. Okay. Simply put, what do you mean by homologous pair? It's a pair of chromosomes. One you got from your father, one you got from your mother. So now listen, before meiosis starts, there's going to be your prophase, no? Prophase. Sorry, prophase, no, interphase. I'm sorry. Before meiosis starts, there's going to be an interphase where there's going to be G1, S, G2. In the S, what's going to happen? DNA copying or DNA replication. So this mother's chromosome is going to get what? This mother's chromosome is going to get a sister, no? The mother's chromosome is going to get a sister because of what? Because of DNA replication. So, in a homologous pair, who else is there? There's the father's, no? There's the father's chromosome is also there. So, the father's chromosome is also going to get a sister because of what? DNA copying. So, now if you take after S phase of interphase, this is what a homologous pair looks like. Hari? Then you can label, you can label these two reds, you can label these two as maternal sister chromatids. They are the same thing, but because of the DNA replication, everyone has gotten a sister. Right? They are maternal sister chromatids, and then of course you have maternal. These are paternal sister chromatids. Maternal sister chromatids and paternal sister chromatids. Okay. Okay. So that is something also. So I know these are simple concepts, but part of you forget why is there 4 chromosomes? Why chromatids in this? Why is there 4? That is because if you when you are making a homologous pair, you have a paternal and a maternal one, but they are also doubled. So altogether you have 4 chromatids in a homologous pair before meiosis starts. You have 4 chromatids in a chromosome pair before meiosis starts. Right? Okay, we jump back to the tute. Now we are going to start properly steps of meiosis. Okay. Before that, yes. So prophase 1 I can explain here. Okay. Right. So we'll write down the things that are happening in prophase 1 first. Hari. We'll take it step by step. We can't grab everything at the same time. No? That's how you get confused. Okay. So in prophase 1, what's going to happen is like this. So there are some things you need to write down here. So the space management is going to be a bit important. Okay. So let's write down. First, of course, you can see so you can see in these stages you have properly condensed chromosomes. Right? You can see they are very compact and nicely packaged. So here you can still see the DNA, the chromosomes are still a bit elongated. No, that means they are still condensing. They are still condensing. Okay, so that's why we have to write down in prophase. We better write down in prophase 1, or we are still in prophase 1. We can write down first point, chromosomes begin to condense. Write with me. Okay, chromosomes begin to condense. Okay. So keep in mind, we are going to take our diploid number as 6. That means if your cells have 46, this cell has only 6. Right? Just to keep it simple. First point, chromosomes begin to condense. So what else can you see? Yes, what else can you see? You can see, so this is a maternal chromosome, no, with the 2 sisters. You can see this is a maternal chromosome with the 2 sisters. Then this is a paternal chromosome with the two sisters. Now what has happened is the maternal and paternal homologous pairs have matched up. So you can see if you take this red and this blue, they are having the same length, no? They are having a similar length and they look similar in their shape, and their centromere is also kind of located in the same level, no? May I get centromere is kind of here, centromere is kind of here. So you can see this red and this blue are homologous chromosomes. This red and this blue are homologous chromosomes. That's why they are matched up. Okay. So we'll write down the steps, then I'll explain. Okay. Next step. What's going to happen next? Yes, what's going to happen next? Nucleolus begin to disappear. So you don't need the nucleolus again, right? You need space. Okay. So the nucleolus disappears. Nucleolus begin to disappear. Okay. Then something important happens, right? Listen. In prophase 1, we are going to do a bit of a mixing of genes from your mother and father. We are going to do a mixing of genes from your mother and father. That is going to be done by cutting the maternal and paternal chromosome and exchanging the pieces and then reconnecting. Alright. So now what's going to happen is in prophase 1, your chromosomes are going to look like this. You can see there's a paternal blue pair, and behind that you have the maternal blue pair, red pair. No? Then what's going to happen is, okay, this maternal and paternals are going to sort of connect with each other, and then part of the maternal is going to come to the paternal. Can you see? Then if you zoom in further, you can see this is a red one, no? This is a red chromosome, no? This one is a red chromosome. Now what has happened to that red chromosome? There is a piece of blue connected to that red chromosome. Yes. Then again on this end also, this is a red chromosome. This is a piece of blue connected to that. Alright? That means maternal genes are going to the father's side, father's genes are coming to the maternal side. So this entire complex is called as a synaptonemal complex. That means they are connected tightly by some special proteins that are going to facilitate this exchange. Right? So that is called the synaptonemal complex. If you take a electron microscope image, you can see something like this, even visible under the electron microscope. Okay. So that is why it's given in your resource. Shall we read the resource? We didn't read the resource. No? Alright, let's read the resource. Wherever nucleolus begin to disappear, we read about that. Next, the formation of a zipper-like structure called the synaptonemal complex by specific proteins holds the two homologues tightly together. Can you highlight a zipper-like structure called the synaptonemal complex? A zipper-like structure called the synaptonemal complex. That is very important. Then the pairing and the physical connection between the homologous chromosomes is called synapsis. Synapsis means this connection between maternal and paternal pair, that is synapsis. That's a definition. Can you highlight? Pairing and physical connection between the homologous chromosomes is called synapsis. Okay. During synapsis, part of the DNA molecule of non-sister chromatids of paired homologous chromosomes break, exchange and reform at the corresponding points. Okay. Now what's going to happen is non-sisters. I'll jump here. Now we are going to subject these to the process of what we call as, crossing over. We are going to subject them to a process called crossing over. Crossing over is like you are sort of cutting DNA, a chromosome, and then giving it to the other person and gluing it together. Right? Okay, so when you subject this to crossing over, when you subject this to crossing over, it's going to look like this. So you need to know who are your non-sisters. So blue are sisters, red are sisters. crossing over is going to happen between non-sisters. That means it has to happen between red blue, blue red. Right? It has to happen between maternal and paternal only. So after crossing over is done, it can happen to so many amounts. So we usually say it happens about 20 times between two homologous chromosomes, but we can't draw that many crossing overs. No, so we'll just draw. Imagine you have this. This is the red chromosome, okay, and this is its sister, maternal one. Okay, then this is the paternal. Sorry, this is the paternal homologous. This is the pat. I have run out of blue ink. Okay, this is the paternal homologous chromosome. Okay, and so after crossing over is done, it's going to be something like this red chromosome. This red chromosome is going to have a piece of blue. Then this blue chromosome can have a piece of red. This blue chromosome can have a piece of red. So mother and father's DNA have exchange. Then what else can happen? I'm just taking some examples, okay? Just taking some examples. You can say in this non-sister chromatid, we have a piece of blue has been connected, okay? So in this red chromosome, now there's a piece of blue. In this blue chromosome, now there's a piece of red. This is called as crossing over. So it's simply an exchange of DNA between the maternal and paternal chromosomes. So because there has been some exchange, we can see the places this took place. We can see the places this took place. So you can call this kind of places where crossing over took place by a special name that is called chiasmata. Right, chiasmata. Chiasmata kiyanne. Okay, it's chiasmata. Okay, chiasmata. Okay, now I think all clear. We can come to the tube and we can complete it. Okay, so you can see, so the places where this crossing over took place, we call it as chiasmata. So they are actually visible under the electron microscope. Okay, right. So we will come to the phase 1 and let's complete the note. Okay, write with me. Okay, write with me. Synaptonemal complex is formed. Synaptonemal complex is formed. Write with me. Okay, third point under phase 1, synaptonemal complex is formed. Then keep writing. Crossing over takes place. Crossing over takes place between non-sister chromatids, of the paired homologous chromosomes. This is very, very important. Between what and what crossing over is happening. Then the critical things are over. Then there is going to be the normal things that are going to happen during a phase 1, right? Such as what? Nuclear envelope is going to break. You can't do anything with a nuclear envelope, no? Right? Nuclear envelope breaks. Then what else? Centrosomes start to produce the spindle. Hurry, you need a spindle, no? You need to move chromosomes here and there. Centrosomes form spindle. Many things are going to happen. Phase 1 of meiosis is such a big phase. Right? So many things are going to happen. Centrosomes form the spindle. Final point, kinetocomicrotubules. I'll write over this side, but you keep it nicely bit under Professor Anhari. Kinetocomicrotubules are attached to each homologous chromosome. Kinetocomicrotubules are attached to each homologous chromosome. If you couldn't write with me, I'll read. Chromosomes begin to condense, nucleolus begin to disappear, synaptonemal complex is formed. Next, crossing over takes place between non-sister chromatids of the paired homologous chromosomes. Nuclear envelope breaks, centrosomes form the spindle. Kinetocomicrotubules are attached to each homologous chromosome. So while you're writing, keep in mind what we are going to do in meiosis 1 is we are going to separate the blues from the reds. We are going to separate the maternal from paternal. Metin, can you see in anaphase 1 you are going to be separating the blue from the reds, reds from blue, blue from the reds. That's what we are going to do. That's why you need kinetocomicrotubules attaching to the red and blue separately. Because why? You have to remove the red's venom, blue's venom. So, the kinetocomicrotubules are attaching to the homologs. Okay. Okay. Any questions? Anyone? You can ask anything, okay? Okay. Right, so big part is over. Meiosis is almost over when you can understand prophase 1. Okay, meiosis is done when you are done with prophase 1. From there onwards, it's easy going. Okay, so we'll move on to metaphase. So, what's happening in this diagram? You can see in our prophase diagram, you can see nuclear envelope is fragmenting, homologous chromosomes are connected, and you can see chiasmata where this crossing over is happening, and you can see the spindle is being formed by the centrosomes. So, all the things we wrote down are visibly happening, no? Okay. So, we move on to metaphase. right? Now in metaphase, it's very similar to your mitosis. The chromosomes are the homologous pairs actually are going to get arranged on our metaphase plate. Can you highlight metaphase plate? Can you highlight metaphase plate? So that's the first thing that's going to happen. Let's write down under metaphase. Okay. Homologous chromosome. Now keep in mind the maternal and paternal chromosomes are still connected. Homologous chromosomes arrange on the metaphase plate. Homologous chromosomes arrange on the metaphase plates. Now keep in mind, if you zoom into this, you can see both threads are connected to a single microtubule coming from one pole, and you can see both blues are connected to a microtubule coming from the other pole, because what we are going to do is we are going to separate the reds from the blues. I keep on saying reds from the blues because maternal and paternal you can visually see, you can see red and blue, no? Okay, so that is such an important thing, because in meiosis 1, what are you going to separate? You are going to separate maternal and paternal. So next point, write down both sister chromatids of a homolog are attached to a kinetochore microtubule from one pole. Kinetochore microtubule from one pole. Both are going to go together. Therefore, they are going to be connected. The sisters are not yet separated. Sisters them are not separated. Okay, only the maternal and paternal are going to be separated. The sisters will not be separated yet. Sisters are not yet separated. Okay. Okay. Right. So we move on to anaphase. Anaphase is always a phase where separation happens. No sisters leave each other. So now what are going to happen is not sisters are leaving each other. Who are going to be leaving each other? The homologous pairs are going to separate. That means mother and father chromosomes are going to separate. Right? Mothers and fathers chromosomes are going to be separated in anaphase 1. Okay. So let's write down what are the things happening. As usual, the microtubules that are attached to the kinetochores, they are going to get shortened. They are going to pull the chromosomes towards the opposite pole. So they are going to get shortened. So you can say, you can write down under anaphase 1, kinetochore microtubules shorten. Kinetochore microtubules shorten. Very important point, put a star mark and write down. Homologous pairs separate and one chromosome of each pair moves to opposite pole. Very important point. Okay, I run out of space. Keep another, put a star mark and write down. Sister chromatids of each chromosome, sister chromatids of each chromosome remain attached. Sisters are not yet separated. Sisters are not yet separated. Each chromosome remain attached. Sister chromatids of each chromosome remain attached. Okay, so if I zoom out a bit again, okay. Anaphase 1. Kinetochore microtubule shorten, homologous pairs separate. One homologous pair separate and one chromosome of each pair move to opposite pole. Then very important, sister chromatids of each chromosome remain attached. Sister chromatids of each chromosome remain attached. Okay. Okay, anyone still writing? Okay, right. Then we move on to telophase and cytokinesis. Now keep in mind, at the end of each meiotic division, that is at the end of meiosis 1 as well as of course at the end of meiosis 2, you are going to have an individual telophase and a cytokinesis. So, telophase 1, there's going to be a cytokinesis, because at the end of meiosis 1, you are going to have 2 cells. Then those 2 cells are going to have their own meiosis 2s. That's how you get 4 cells, right? So, if you go to the bottom of this, you can see we are going to be following the 2 cells that you got at the end of meiosis 1. So, that you are following 2 cells at the same time. That's how you get at the end 4 cells in total. Ne? That's how you get in the end altogether 4 cells. Okay, that is because at the end of meiosis 1 there is a cytokinesis. Cytokinesis means what? A division of the cytoplasm. Cytokinesis. Okay, cytokinesis so telophase 1 is nothing much. Let's read. All the normal things that you have happening at the end of a division, what are the things? We can say telophase 1 again, what are the things happening? Nuclear envelope reforms. So, write with me, nuclear envelope reforms. Then nucleoli reappear. Nucleoli reappear. Spindle disintegrate. Spindle disintegrates. Chromosomes decondense. Write with me. Chromosomes decondense. Finally, genetically non-identical. Why are they genetically non-identical? Why are they genetically non-identical? You can see because of crossing over, these reds have pieces of blue, these blues have pieces of red, these blues have pieces of red, this red has piece of blue. So, the cells that are being produced at the end of meiosis 1 are already genetically non-identical. Because why? Crossing over, simply because of the crossing over. Genetically non-identical, haploid, two daughter cells, cells are produced, but you have to say cells are produced after cytokinesis. You get cells only after cytokinesis. After cytokinesis, right? So, I will read again. Nuclear envelope reforms, nucleoli reappear, spindle disintegrates, chromosomes decondense, genetically non-identical haploid daughter cells are produced after cytokinesis. Okay. What happened? Did I get disconnected? No, right? How did I? Did I get disconnected? What? That was very sudden. Yes. Okay. I left the meeting. How's that possible? Okay, there's a virus. Okay. Okay, done with telophase 1. Okay. Okay, so now what we have to simply, now the difficult part is over. I keep on saying that, right? Yes, once you learn something, the difficult part is over. There's more things to be learned, right? We move on to meiosis 2, right? We move on to meiosis 2. Similar to meiosis 1, you are going to have prophase, metaphase, anaphase, telophase, and cytophase. So you might be thinking, why is there no pro-metaphase? Why is there no pro-metaphase? Actually, pro-metaphase is something that recently was introduced into this our study of biology. I mean, into the international study of biology, because they found out, okay, particularly during certain drug development, certain antibiotics development, particularly not antibiotics, sorry, certain actually anti-cancer drug development, they actually went a lot deeper into cell division, because certain anti-cancer drugs are designed to inhibit certain steps of certain phases of the cell cycle. So when they did that, they found out a lot about each of the phases, that more than we study here, definitely. So that's why they introduced a mitotic phase, that is between prophase and metaphase, as prometaphase. So in meiosis, we don't actually talk about a prometaphase, because we are not that concerned with each of the individual steps as much as in mitosis, because in mitosis it's important that we design certain anti-cancer drugs that are able to sort of inhibit specifically a certain phase. Okay, clear? So that's why we don't have a prometaphase in meiosis. Okay, let's see what's going to happen in prophase 2. Okay, I'll clear up the diagram so that you can see. Prophase 2, meiosis 2, I told you, it is similar to mitosis, right? Meiosis 2 is similar to mitosis. Why do I say that? Meiosis 2 is similar to mitosis. It is because simply in mitosis, what's happening? You are separating sisters. In meiosis 2 also, what you are doing is you are separating sisters. Okay. So we'll talk about prophase 2. Prophase 2 what are the things that's going to happen? Let's write down. Again you start a cell cycle. Again you start a mitotic division. What are the things you need to do? So there's a little space problem, but try to manage it. Nothing much to be written. Centrosomes produce spindle. Chromatin condense into chromosomes. With two, very important, with two unidentical sisters, because now the sisters are weird. The sisters were mixed up. What happened to the sisters? The sisters are no longer identical, because if you can zoom in, you can see here there's a sister. These are the two sisters. Okay, these are the two sisters. This is the red sister. This is a red chromosome. So there's a completely red one, and there's another red one with a what? With some blue. With some blue. So are the sisters identical now? No. Right? Then there's a blue one here. There's a blue one not undergone crossing over. Then there's the other blue sister. What has happened to that blue sister? There's a piece of red in that blue. Okay. So even though they are sisters, they are now unidentical sisters. So that's the most interesting thing about prophase 1 as well as in meiosis 2, prophase 2, because the sisters you are talking about here, unidentical sisters. Okay. Chromatin condenses into chromosomes with two unidentical sister chromatids with two unidentical sister chromatids. Then next one, of course, the normal things that are happening, nuclear envelope breaks, blah blah. Then nucleolus disappears. Nucleolus disappears. Centrosomes produce the spindle. Chromatin condenses into chromosomes with two unidentical sister chromatids. Nuclear envelope breaks, nucleolus disappears. Okay, so everyone listen while you are writing. Tonight itself, I will be sharing the beginning of our metabolism. That means metabolism introduction as well as enzymes first day recording into the groups available for you free of charge. Okay, so I want you to watch that, start watching that. It's a 4-hour recording before the exam. Okay, if you didn't join at the beginning of enzyme, I want you to watch that recording and complete the tunes. Okay, so with that you get a thorough understanding of enzymes, then you can face the paper. The first thing I want to tell you about the paper, don't stress about it. Just study as you study your O-level science a little bit, come to the paper and see what happens. See what happens. This is the time to see what happens. This is the time to see what happens. You can go to a class and see what happens. You can write a paper and see what happens. This is the time to experiment. Right? Not in another year's time. This is the time to experiment. Okay, you can go to different classes and see if you are liking the vibe of the class. If you don't go there. Okay, you have the time to test things, not in another year's time. Okay. Right. Prophase 1. Sorry, prophase 2. Done? Prophase 2. Then metaphase, right? So, of course, with any metaphase, what is the most important thing about a metaphase is the metaphase plate. Most important thing about a metaphase is the metaphase plate. You can see now, you can compare the metaphase 2 with metaphase 1. Can you see metaphase 2 and metaphase 1? The metaphase 1 plate, metaphase plate was, if it was horizontal, metaphase 2 metaphase plate is going to be vertical. Somehow, if metaphase 1 plate was vertical, metaphase 2 plate is going to be horizontal. Always meiosis 1 metaphase, meiosis 2 metaphase plates are going to be perpendicular. Vertical is going to be horizontal. If the other one is horizontal, this one is going to be vertical. Always occurs in the perpendicular direction to each other. That is actually the most important thing you need to know about metaphase 2. Okay, so let's write down about metaphase 2. I'm not going too fast neither. Yes. Write down centromeres of all chromosomes arrange at metaphase 2 plate. Star mark can write down, put a star mark and write down. Metaphase plate of meiosis 2 is perpendicular to metaphase plate of meiosis 1. Metaphase plate of meiosis 2 is perpendicular to the metaphase plate of meiosis 1. Okay, I should tie. Okay, there's only a small amount. So metaphase 2, what the only thing you need to know is it's going to happen in the perpendicular direction of metaphase 1. Okay, anaphase 2 is remaining. So what's going to happen if we zoom out? It's, I told you, it's similar to what is happening in mitosis. The sister chromatids are going to separate. But the special thing about these sisters, is that what? The sisters are unidentical. This blue sister has some red. This sister is a complete blue. This sister is a complete red, but this sister has two blues. Okay, the sisters are now unidentical because of what? Anyone? Why are they unidentical? Why are they unidentical? Why are they unidentical? Why are the sisters unidentical? Because of yes, crossing over. Yes, good. Okay, because of crossing over. Let's write down anaphase 2. I'll use red color. Sister chromatids are separated. Sister chromatids are separated at the centromere. Sister chromatids are separated at the centromere and they become chromosomes. So once the chromatids are separated, they are known as chromosomes. Due to shortening of kinetocomicrotubules, they move towards opposite poles. They move towards opposite poles. Okay, this is what's happening in anaphase 2. Quickly write down Simply a separation of sisters. Simply a separation of sisters. Okay, all right. Okay. Telophase and cytokinesis is remaining, and at the end we are going to do a small comparison between mitosis and meiosis, then we are done. Okay, right. Normal things happening in telophase. Now by this point you should be able to predict, okay, if you are a scientist discovering telophase, you should be able to, I don't need to even look under the microscope to say what is happening here. You can simply predict, right? The chromosomes decondense to what? To chromatin. Chromosomes decondense to chromatin. Nuclear envelope and nucleolus reforms. Put points, these are all individual points. Spindle, sorry, disassembles, spindle disassembles 4 genetically, final point, put a star mark and write down, 4 genetically non-identical haploid daughter cells are produced after cytokinesis. Chromosomes decondense into chromatin, nuclear envelope and nucleolus reforms, spindle disassembles, at the end 4 genetically non-identical haploid daughter cells are produced after cytokinesis. If you are going to have cells, that is going to be only after a division of cytoplasm, which we call as cytokinesis. Okay, that is going to only happen after a cytokinesis. If cytokinesis doesn't happen, you have simply a telophase. What are you going to have? Genetically non-identical daughter nuclei only, no cells. Okay. Okay. Right. Next, we are going to do the final conclusion of our lesson. That is, we are going to do a comparison between mitosis and meiosis. Okay, between mitosis and meiosis. With that, cell division, cell cycle is over. We are going to compare DNA replication, how many times it occurs, number of divisions, is there any synapses, the number of daughter cells and what is their genetic composition, what is their role in the animal body. Okay. Okay, let's write down. In the mitosis, can you see it occurs in both diploid and haploid cells? There's a story that I don't want you to think about a lot, because there's in the lesson plant reproduction, that is in our fourth lesson. Actually, no, I'm going to be doing that in with your third lesson. With the evolution and diversity lesson, I'm going to be doing our plant reproduction as well. So you can see this, all these pages are related to plant life cycles, plant reproduction. Okay, so plant reproduction is going to be done in your third lesson. So there you will learn that in plants there is a generation that is haploid. Again, there's a type of plant in their life cycle that is haploid. Don't overthink about it. So in that generation there is mitotic division. So that's why we say mitosis can occur in both diploid and haploid, because simply what mitosis is doing is it is copying any cell that is given to him, right? If you want to subject a cell into mitosis, it doesn't care. He's just going to copy it exactly it is. It's a photocopy machine. Meiosis is not like that, no. Meiosis does some crossing over, then the homologs are separated. So many things are done. Therefore, meiosis only takes place in diploid cells. Meiosis only takes place in diploid cells. Keep that in mind. Okay. DNA replication number, you can write here on the mitosis side. I'll use the same sort of green color, or yellow color. Occurs once in once in S phase of interphase. Occurs once in the S phase of interphase. That is the only time where you have DNA copying. How many times DNA are copied in meiosis? How many times are DNA copied in meiosis? How many times DNA are copied in meiosis? Before meiosis or during meiosis, whatever. How many times? Yes. Still once. In meiosis also occurs once. Only one copying. Occurs once. Before, write with me, occurs once before meiosis 1, but not before meiosis 2. There's only DNA copying before meiosis 1. There is no copying in between. Occurs once before meiosis 1, but not before meiosis 2. But not before meiosis 2. Okay, not before meiosis 2. Number of divisions. Number of divisions. I want you to list down the divisions here. I'll only write the short form. Hurry. There's prophase, there's prometa, there is ana, there is telo. There is prophase, prometaphase, prophase, prometaphase. I missed one. There's metaphase, sorry. Prophase, prometaphase, metaphase, anaphase, and telophase. In meiosis, okay, there are two divisions. 2, each with prophase metaphase, anaphase and telophase. In mitosis, number of divisions: prophase, prometaphase, metaphase, anaphase and telophase. In meiosis, there is 2, each with prophase, metaphase, anaphase and telophase. Synapsis of homologous chromosomes, that's the second thing. So I have sort of come down a little bit. Okay, so try to match the space. Synapsis of homologous chromosomes doesn't occur in mitosis. I write the same thing. Number of daughter cells 2, genetically identical. Okay, number of daughter cells is 2, they are genetically identical. Then on this side you can say synapsis of homologous chromosomes occurs during prophase 1. Synapsis occurs during prophase 1. Then number of daughter cells, you have to say 4, each is haploid and genetically unidentical. 4, each is haploid and genetically unidentical. Okay. Then the role in the animal or plant body කියනකට there is a significance of meiosis කියලා there's in a resource book. Okay, there's a list of things called significance of meiosis, such as maintains the constant number of chromosomes, produces new genetic variations. Okay, so those things you can write here in the meiosis side. In the mitosis side you can write down asexual reproduction, genetic composition, maintains genetic composition. Okay, cell regeneration, tissue repair, those kind of things you can write down in the mitosis side. Okay. Okay. So today we covered, we went from the beginning almost of mitosis, then tumor, cancer and goals, and we completely finished meiosis. Okay, so now what I want you to do is, I have sent the recordings of beginning of metabolism as well as the start of enzymes. I want you to watch that recording if you have missed it before you do the paper. Okay, so with that I think we can conclude our up to speed sessions. We have done 6 of them now. Okay, almost 4.5 hour sessions, 6 sessions of them. We went from the very beginning of introduction to biology and up to the end of cell division within 6 sessions. So at the same time I was solving issues, I was clarifying doubts, and as well as I was showing you some extra things. otherwise you guys are going to get bored. So I think we managed to cover my sort of a satisfactory portion within this 6 sessions. Okay, so I hope you got the maximum benefit out of those. You can share the playlist, okay, and for any friend of yours who are seeming to be lost with the first few lessons of the syllabus, and yeah, you can help them out. They are available in my YouTube channel. Okay, so we can wrap it up. Okay, so I hope any sort of, let's say, low confidence that you had, that you join later or you missed this section again, all those doubts were clarified. My main message for you is, do not overthink at this point, but see and try what you like and what you feel like is the correct thing, correct path to follow. Okay, so if you feel like, as a sensible person, this is the way I am going to get my best result, go in that pathway. I am going to help you to the 100% that I can. Okay, so up to speed sessions are over. We'll finish it, neither. Okay, so any issues that you want